Hello friends, I'm Shruti Parashar. Welcome to my channel Biology 360. Today I'm going to discuss the topic what are enzymes and the properties of enzymes. The topic is important for ICSE, CBSE, IGCSE and HSC board students and for students preparing for NEET, Olympiad exams and AIMS exam. Enzymes. Each enzyme catalyzes a small number of reactions, specifically perhaps only one. So enzymes are they are also called as biocatalyst. These enzymes, they speed up the rate of reaction and at a given time, they can catalyze only one reaction. The substance upon which an enzyme acts is termed as the substrate. So substrate can be a glucose molecule or it can be a sucrose molecule or it can be a protein molecule as well. Now there are two main types of enzymes. One is called as endoenzymes and another are the exoenzymes. An endoenzyme or intracellular enzyme is an enzyme that functions within the cell in which it was produced. And an exoenzyme, or it is also known as extracellular enzyme, is an enzyme that is secreted by a cell and functions outside that cell. Exoenzymes are produced by both prokaryotic and eukaryotic cells and have been shown to be a crucial component of many biological processes. On the basis of chemical composition, enzymes can be put into two main categories. Number one, purely proteinaceous enzymes. These are the enzymes which are purely proteinaceous in nature. Example, proteases enzymes that split protein molecules. And second category of enzymes are the conjugated enzymes. These are made up of a protein to which a non-protein prosthetic group is attached. There are enzymes which require certain organic compounds and inorganic ions for their activity. The organic compounds that are tightly attached to the protein are called coenzymes, whereas the inorganic ions which are loosely attached to the protein part are called cofactors. Now let us discuss the properties of enzymes. First, proteinaceous nature. All enzymes are basically made up of protein. Second, three-dimensional conformation. All enzymes have specific three-dimensional conformation. They have one or more active site to which a substrate or reactant combines. The points of active site where the substrate joins with the enzyme is called substrate binding site. So to explain this point, I have a diagram in the next slide. So all the enzymes, they have a specific site on their structure and that site is called as substrate binding site. So the substrate binds to the enzyme at the substrate binding site and they both form enzyme substrate complex. Catalytic property. A small quantity of enzyme can catalyze the transformation of a very large quantity of the substrate into an end product. So, enzymes are required in very minute amount to catalyze any given reaction. Specificity of action. Each enzyme acts upon a specific substrate or a group of specific substrates. So, enzymes are very specific in their action. They will act only upon a specific substrate. Say for example, lipase enzyme, they will act only on lipids. Proteases enzyme, these enzyme will act only on the proteins. So enzyme, they are very specific. They act on only specific substrate. Each enzyme exhibits its highest activity at a specific pH called optimum pH. Fifth characteristic, temperature. Most of the enzymes work at an optimum temperature between 20 and 35 degrees Celsius. So enzymes are very active in 20 to 35 degrees Celsius temperature range. Enzymes are destroyed at higher temperature of 60 to 70 degrees Celsius or below. So very high temperature or very low temperature will not help enzymes to function efficiently. They require an optimum temperature range. In this slide, we see the enzyme function. There is an enzyme and the substrate binds to the enzyme at the substrate binding site and they form enzyme substrate complex. Now the enzymes will act on the substrate and substrate will get converted into a product, forming enzyme product complex. And later the products are released from the enzyme. In this diagram, we see a protein portion of an enzyme. It is called as apoenzyme. It is an inactive region. Plus, we see a non-protein portion which is called as coenzyme. So, coenzyme is acting as an activator here. So, when coenzyme will get attached to the apoenzyme, it is called as a holoenzyme. And now this holoenzyme or the whole enzyme is very active. The substrate will bind to the enzyme and the substrate will get converted into products. Friends, do subscribe to my channel Biology 360 and don't forget to click on the bell icon to receive the notifications on the biology videos. Also, please send me your comments and suggestions regarding the video as well. Thank you.